This video will review the organization of the cerebral cortex. Here we're looking at the dorsal surface of the brain, and what we see separating these two hemispheres is a longitudinal fissure. Now you can get a really good feeling here for those gyri and those sulci, the hills and the valleys here. Here's the central sulcus that separates the frontal lobe from the rest of the brain. And we'll get into some of these divisions in our next video. Here's a horizontal section of the brain and showing you the ventricles that we'll talk about momentarily. Right here that contains cerebral spinal fluid. Here's the front of the brain or the frontal lobe. This is the most anterior portion. Here's a posterior portion. Here's the thalamus, again, sitting nicely in the center of the brain. Here's the basal ganglia. And here's an area that we're going to talk about, the corpus callosum that connects these two hemispheres. Because you see here, when we get up into this cerebral cortex and those subcortical structures, we have two hemispheres, two complete hemispheres that are separate. And so we need those hemispheres to communicate quickly, and they do that with the corpus callosum. So as we just said, cerebral cortex is divided into two hemispheres, a left hemisphere and a right hemisphere. So it's divided into two halves. Now this forebrain is separated into these two halves, the left hemisphere and a right hemisphere, and they're doing different things, we want those to be able to communicate very quickly so that our actions, our movements, our behaviors can be fluid. So they're connected with what's called the corpus callosum. And the corpus callosum is a bundle of axons that's myelinated because we want that communication to be quick. In addition to the corpus callosum, we also have what's called the anterior commissure. Because if you notice here, the corpus callosum runs most of the length of the brain. However, in the very anterior portion, it does not. And so we have and there's a few other smaller ones also, but again, they're myelinated, bundles of myelinated axons that connect the two hemispheres. And so the corpus callosum is the main one, and that's something that you'll need to label in your handout. The anterior commissure is not represented there. So each side of the brain, or hemisphere of the brain, receives sensory information from the contralateral side. So sensation of touch on your right arm is going to travel to your left somatosensory cortex, so the left hemisphere of your brain. Information from your skin in the left side of your body, so sensation of touch when you touch something prickly with your pointer finger on your left hand, that information is going to go to the right hemisphere, the somatosensory cortex in the right hemisphere, the right side of your brain. So that's the contralateral side, the opposite side of the body. Each side also controls motor movement from the contralateral side. So your motor cortex that we're going to talk about in a little bit, that's on the right side of your brain in the right hemisphere, is going to control information on the left side of your body. So those neurons in the motor cortex of your right hemisphere control your fingers on the left side of your body. That's the contralateral side, or the opposite side. The same side would be ipsilateral. But important to understand that that sensory information comes in and crosses and goes to the opposite side of the contralateral side of the brain. Motor control from one side of the brain is going to control information or movement on the opposite side of the body, the contralateral side of the body. This is just showing you our brain in relationship to other brains. Here's the human brain. You can see all those hills and valleys, those gyri and those sulci. You can see a chimpanzee, a little bit smaller brain, but has many of the same gyri and sulci. Zebra has a lot of these as well. A raccoon, much smaller brain, but still has some of these hills and valleys. But then we get to a hedgehog and we get into a manatee, and we don't see that. So that means that even though the manatee has a brain that's not the same size as a chimpanzee, it doesn't have that same surface area. So it doesn't have the same number of neurons. So now we're going to discuss the organization of the cerebral cortex. And the cerebral cortex contains up to 6 distinct laminae, or layers. And those are parallel to the surface of the cortex. So again, kind of take your hand and put it out in front of you. It's like, this is what I would do in class. And you see you have one, then put your other hand below it. It's laying horizontally. You have 1, 2, 3, 4, 5, 6 layers that are all parallel to the surface of that cortex. And so this is shown here. And so here are those 6 layers. depicted as Roman numerals. What you'll notice here is that not all the layers are the same size in the different cortex. So in the primary motor cortex, layers 5 and 6 are much bigger than you see in the primary sensory cortex. And I'm not going to go into the ins and outs of these, but just know that in the different layers, different neurons have different functions. So we have the 6 distinct laminae, or layers. We also have columns that lie perpendicular. So if you were to take your hand and then take like a toilet paper roll and put that up against the bottom of your hand, those are the columns. And so that within that column, you're going to have each of those 6 layers. So again, those columns appear to have separate and distinct roles as well. So here's it showing you, here's one column, here's another column, here's another, and so on and so forth. And then within those columns, you have the layers. And so typically the axons of the neurons in a given column are going to stay within that column, projecting down and having inputs coming up into it. So something else I want to introduce to you here is this idea of ventricles. Mentioned it earlier. So you see these, they kind of look like holes. And I actually got yelled out by our ultrasound technician when they were doing ultrasound on my oldest child, Carrie. She was looking at the brain, and I was all excited to see the brain on the ultrasound. And we're looking, and I saw the ventricles, and I said, "Oh, are those little holes? Are those the ventricles?" She said, "They are not holes. They are fluid-filled cavities." I said, "Well, I know that. I just wanted to know if they look like holes. Are those the ventricles?" And she said, "They are not holes." So anyways, I always think about that when I talk about ventricles in the brain. So they are four fluid-filled cavities within the brain, and they contain cerebral spinal fluid. Now, cerebral spinal fluid is a clear fluid that's found both in the brain and the spinal cord. In the spinal cord, we have the central canal, and it acts as a cushion for the brain, so it provides a cushioning for the brain, so that, again, you can have some minor head trauma, and you still have this protection for the brain. And the other important role that it does is it provides a reservoir of hormones and nutrition for the brain and spinal cord. It also helps flush out what I call my non-scientific term, the gunk of the brain. So it helps clear out things that we might not want to stay in the brain as well. And so it's got a very important role. And what you'll see happening, and we'll see some of this in some diagrams later in the semester, our images, is that when you have brain damage, so if you have an area of the brain that dies, these ventricles get larger, and so that cerebral spinal fluid just kind of fills in, so you don't have any gaps in the brain, and so it just kind of maintaining that cushioning. But so when you see enlarged ventricles in a person's brain, that means that they've had cell death somewhere in their brain. So this is again just showing you those ventricles. The blue is the lateral ventricles here. This is the third ventricle in the center of the brain, and then here going down to that central canal of the spinal cord. And here's just a cross section again showing you what that would look like. Again, I've always described it as a hole, but the ultrasound technician didn't like that.